Good morning, everyone. This is super exciting. Thank you for being here in person. And we also have a virtual audience today. This is kind of a new thing we're trying. I'm really excited about it. And I'm really pleased to have the T32 supported junior scientists. I don't like to call them trainees. Come here today and give us flash talks on their projects. And we also have the external advisors visiting us today, Dr. Channing Durr and Dr. Carrie Bernstein. And we'll have a day of talking about our T32. And so I thought it'd be fun to feature our trainees that are supported by the T32 as we enter our the end of our cycle, which will be 50 years of funding by the NIH. So I'm really excited about that. And so I will introduce each speaker. They'll have 3 minutes to tell you all the exciting things about their project. And we'll have a couple minutes for a question or two after each. And we've got 8 today. So we've got plenty of time to do this. So without further delay, our first pre-doctoral candidate speaker is Joel Pardo from the Yee Lab, examining the role of ApoBec3B in the development of breast cancer treatment resistance. Thanks, Joel.
Thank you, Dr. Lang, for the introduction. So yeah, I'm Joel. I'm working on figuring out what the role of ApoBec3B is in the development of breast cancer resistance. And so I guess first off, what is ApoBecs? And so ApoBecs are this group of enzymes that essentially endogenously are thought to restrict viruses, so essentially mutate viruses and lead to essentially inactivating the virus. However, they can also act on endogenous DNA and essentially mutate endogenous DNA and lead to the formation of cancer. And so the question then is, what role does it have beyond just the formation of cancer? And there's been evidence that ApoBecs play a role in tumor evolution and tumor heterogeneity and how these tumors evolve to become resistant to different treatment modalities. And so in sort of the middle panel, you can see that treatment with patients with luminal A tumors that received treatment with tamoxifen, they had overall increased recurrence correlating with higher A3B levels. And based on my initial studies, we see that it turns out that luminal A has by far the lowest level of A3B expression, and it's much higher in the other sort of PAM50 subtypes, namely basal, HER2, and luminal B. And so these are generally thought to be a little more challenging to treat within patients. Going a little further, we see that using some bioinformatics approach, there's a lot of pathways that are upregulated with A3B expression, most significantly those related to cell cycle and immune signaling. And so just highlighted here is one of those pathways, the PD-1, PD-L1 pathway. That is in red are those genes that are upregulated. And so this is just one that actually turns out to be therapeutically sort of treatable. And so then the question is, these are just ways of or just purely correlations. And so the question is, what is the actual causation mechanism? And so to do that, we have made some A3B inducible cell lines that ultimately will allow us to further figure out exactly how that mechanism might be happening, how resistance might be developing these different A3B tumor types. And so ultimately, we hope that maybe we can then use A3B as sort of a prognostic or predictive marker for different treatments going forward. And with that, I'll take any questions.
Thank you, Joel. I just have a quick question. So is it known how the ApoBec3B expression is regulated?
No, that's sort of the short answer. It's not very clear. I think the interesting thing about ApoBec3B is that it's nuclearly localized, and so that's why we're focused on it. So it has direct access to endogenous DNA. As for what upregulates it, I think there's evidence that it's interferon related, but the exact mechanism isn't very well characterized yet.
Carrie?
I don't think I need it.
Okay.
Oh, right. Does ApoBec get activated by exogenous nucleic acids entering the cell?
Yes.
So does it interact at all with STING?
Yes, it might. So the short answer. There's a new, relatively newer publication that shows that ApoBec is upregulated when you give it when there's STING activation. But yeah.
Scott,
So A3B upregulation could increase tumor heterogeneity or it could activate a specific pathway. Do you see evidence in breast cancers whether it could be one or the other? Because I would guess if it was activating a specific pathway, those cancers would be less clonally diverse. But if it was just a tumor heterogeneity mechanism, then those cancers would be more diverse.
Yeah, I think that's a pretty challenging question to answer as it is. So I think we see by far the pathways that are most upregulated are those related to immune signaling. And so that might just be that immune signaling is driving APOBEC expression potentially. And so the question of whether or not APOBEC then is changing it in a specific direction for more resistance is kind of the next objective of with inducible model. But it could just be a general heterogeneity.
Great. Thank you so much, Joelle.
Thank you.
Okay, awesome. Our next speaker is Natasha Roman Ortiz from the Ostrander Lab, and she'll be telling us about targeting breast cancer stem cells with oncolytic retrovirus.
Yep, I just got it. Yep, thank you. Breast cancer stem cells are like the killer bees. Killer bees are invasive, aggressive, and are responsible for maintaining the beehive's population by reproducing. Similarly, breast cancer stem cells can invade other tissues. They can self-renew, and they can sustain tumor growth. Patients that have metastatic estrogen receptor positive breast cancer have a prolonged risk of recurrence, and they have limited therapeutic options due to they have limited therapeutic options. Chemotherapy and endocrine therapy primarily target actively proliferating cells. However, breast cancer stem cells are unresponsive to these treatments due to their slowly proliferative nature. Ultimately, breast cancer stem cells can regenerate a tumor, causing metastasis and breast cancer recurrence. My research entails targeting breast cancer stem cells with oncolytic viruses. Oncolytic viruses are able to cause direct tumor lysis without harming normal cells. My PhD thesis focuses on studying mammalian orthoreovirus, or MRV. MRV has been used in clinical trials for metastatic breast cancer. In these trials, it has been found that MRV is safe and effective when used in combination therapy. However, I aim to create novel MRV strains using a method called serial passaging. But what is serial passaging? Serial passaging is the process that involves making a virus go through multiple rounds of infection and replication in a specific cell type. The expectation is that the resulting viral strains will have superior capacity at killing that cell type. For my studies, I use therapy-resistant tumor spheres. Why? Therapy-resistant tumor spheres enrich for breast cancer stem cells. So what I have found in my research is that the serial passage strains are superior at killing the therapy-resistant tumor spheres. Additionally, I have isolated 3 different viral clones, and out of these 3 different viral clones, there is one clone that specifically robustly kills our therapy-resistant tumor spheres. This data is very compelling because it indicates that the serial passage viruses can enhance a virus' ability to kill. Our ongoing studies focus on characterizing the MRV strains. Ultimately, we want to elucidate the cell death mechanisms elicited by MRV. So, in conclusion, oncolytic viruses can be used as tools to study breast cancer stem cell infection and killing. Oncolytic viruses are promising therapeutic interventions due to their tumor selectivity, ability to induce immunogenic cell death, and their potential to be used in combination therapy. We anticipate that in the future, oncolytic viruses could be able to enhance patient outcomes and ultimately reduce the risk of breast cancer recurrence. Thank you.
Questions for Natasha? Dr. Yee?
Really interesting. Can you say a word about specificity of the virus and how it can distinguish breast cancer stem cells from all other stem cells or cells in general? I guess it's a question of how does it enter the cells and recognize them?
Can you say that last
How,
sentence?
how does the virus recognize cells and enter them?
Yes. So this virus primarily targets cancer cells, and the cancer cells have defective p53. They also have overactive RAS pathway and have a defective antiviral response. And this is why the virus is able to replicate in that environment. Also, the receptor for the virus is JAM-A, and cells that have increased JAM-A expression also are susceptible for the virus infection and killing.
I'm interested in the approach that you're going to use to study that clone B and determine what's different with it relative to the other clones or the parental clone.
Yes. So the approach that we're taking is sequencing the viral RNA. So this virus is a double-stranded RNA virus. So we want to identify what mutations came from that serial passaging. We anticipate that the mutations that we're going to find are going to be either in the attachment protein or in some enzyme related to the replication that can make it efficient, more efficient than the parent virus.
Really cool. There's a question in the back. That'll be the last question for Natasha.
Great job, Natasha.
Thank you.
Your clonal data looks really great. Are you planning on doing any in vivo work to see if that will be like replicated in an in vivo model?
Yes, that is an excellent question. Right now, we don't have plans to do it, but definitely at future directions. This virus, not specifically this virus, but there's another virus, adenovirus, that has been studied in vivo. And it's promising, but for now, we haven't thought about it for our project.
Thank you, Natasha. Excellent.
Thank
Very
you.
good. Our next predoctoral talk is Jose Valentin Lopez from the DAME Lab, elucidating the role of extracellular fatty acids in ferroptosis and castrate-resistant prostate cancer.
Okay, perfect. Good morning, everyone. So as Carl mentioned, I'm Jose Valentin Lopez. I'm a G3 in Scott Dame's lab. And the title of my project is elucidating the role of extracellular fatty acids in ferroptosis in castration-resistant prostate cancer, or CRPC. I'm focusing on CRPC because it is the stage of prostate cancer that's responsible for most, if not all, deaths here in the United States. It is the state of prostate cancer with a very few amount of targeted therapy. So there is a need to improve and define new therapies for this state of prostate cancer. Additionally, I'm studying extracellular fatty acids because there has been a link between diet and prostate cancer progression and aggressiveness. And I'm also studying ferroptosis, which is a mechanism of cell death that's dependent on lipid peroxidation. I'm studying this because it is a way that we could basically link fatty acids to cellular death in prostate cancer. Let me see if I can have a pointer here. I don't think there's a pointer. Oh, there we go. And there's a pointer. Perfect. And ferroptosis is this form of cell death that's dependent on lipid peroxidation, as I mentioned earlier. Polyunsaturated fatty acids are the fatty acids that are more susceptible to lipid peroxidation. So it is necessary to have those fatty acids in the cell for ferroptosis to occur. It has been observed that saturated and monounsaturated fatty acids can be protective against ferroptosis, but polyunsaturated fatty acids can induce ferroptosis in the system. The enzyme responsible for integrating polyunsaturated fatty acids into cellular structures is ACSL4. It has been observed that ACSL4 is more highly expressed in two models of castration-resistant prostate cancer, LNCAP95 here in red, and PC3. And this is relative to the LNCAP model, which is a model of castration-sensitive prostate cancer. Additionally, using mass spectrometry, we evaluated the content of polyunsaturated fatty acids in LNCAP and LNCAP95, and we have also observed that LNCAP95, or basically the CRPC model, contains more polyunsaturated fatty acids than the LNCAP model. And these two things put CRPC models in a more susceptible space for apoptosis to occur in them. And additionally, we also evaluated what was the need for these extracellular fatty acids and their role in possibly inducing apoptosis. So we cultured all three models that I mentioned earlier, the castration-sensitive model LINCAP and the castration-resistant model LINCAP 95 and PC3 in lipid-free medium, which is here in red, and we saw a reduction in growth in all of them. However, when we added back, or when we added Ferrocyan-1, which is an antioxidant, it is a ferroptosis inhibitor. I saw a growth rescue in LINCAP 95 and PC3, which are the two CRPC models. And this result here basically shows that CRPC can be induced in CRPC models, but not castration-sensitive models by removing extracellular fatty acids. So the overall goal of my project is to continue studying ferroptosis and continue studying how it can be induced in castration-resistant prostate cancer as an alternative therapy for this disease. So with that, I'll take any questions.
Great, thank you. Channing.
So this is not my field, so it's a naive question, but I look at these studies and I see some classic prostate cell lines that have been around for decades. And I'm aware that there have been recent developments in organoid models for prostate cancer. Are these still state-of-the-art models, even though they've been around for a while, or would it be perhaps more of interest to transition these type of studies to organoid models that are patient-derived?
Yeah. So right now I'm using these models just to, I guess, like model the transition from a castration-sensitive to a castration-resistant state. I agree with you. It would be best to also continue exploring other models that better resemble patient disease, like patient-derived organoids or patient-derived xenografts as well. But for now, in the first years of my training, these are the models that I've been using for my studies.
Carrie.
It's cool work. Can ACSL4 is it sufficient in LNCAP to make the cell sensitive to ferroptosis induced by polyunsaturated fatty acids?
Yeah, so that's some other work that I'm also exploring and currently working on in the lab. And of course, this is a very short presentation, but I am currently verifying if ACSL4 is the sole enzyme that is responsible for inducing ferroptosis sensitivity in the cell line. So a lot of knockdown and knock-in stuff happening. But at the moment, it seems like it's the main one responsible for this.
Great. Okay, we should move on. Thank you so much. Very nice. Okay, now we're moving into our postdocs. We've got 5 talks from our postdocs. The first is from Yatu Chang from the Nyan Lab, a potential role of alternate splicing control in DNA damage response. Thank you.
All right, thank you for the introduction. So I think everyone can agree that we now live in a very stressful world, and to maintain a healthy life, in addition to all the housekeeping work, we also need an alternative approach to cope with all the stresses. And then it's the same for the cells. All sorts of stresses constantly challenge the cells, DNA damage being one of them. So once DNA is damaged, cell would try to fix them to live, to die, or to die. All of these reactions needs a lot of proteins, and alternative splicing is the way for cells to diversify their protein production. And the alternative splicing is tightly regulated by splicing factors. And why does matter in my research? So our lab is interested in MDS, or myelodysplastic syndromes, which is a group of cancers where immature blood cells in the bone marrow never mature or become healthy blood cells. Interestingly, about 50% of the MDS patients have mutations in splicing factors, and U2F1 is one of the frequently mutated splicing factors. And the hotspot mutation S34F is associated with an increased risk of secondary AML. Therefore, how we are going to treat MDS patients is one of the main questions of my research. Interestingly, it's been shown that U2F1 is phosphorylated upon DNA damage, suggesting that U2F1 may be linking the two processes. And then, so we are interested in whether we can utilize this feature to identify novel therapeutics. To do so, we are going to perform CRISPR-Cas9 genetic screen using a DNA damage focused guide RNA library. We have engineered cells to conditionally express S34F mutations, and then we want to identify the genetic vulnerabilities in these mutant cells. Additionally, our lab has shown that S34F mutants are hypersensitive to PARP inhibitors. Therefore, we are interested in whether we can combine PARP inhibitors with other inhibitors that target DNA damage response pathways. We are hoping that finishing this project will provide insight into how we can better treat MDS patients with U2F1 mutations. Thank you.
Very nice, got you. So is it known which kinase phosphorylates U2AF1?
That's a very good question. It's very little known about U2F1 regulation. So it's a completely new area. So it's not known yet.
Serine or threonine or tyrosine?
Yeah, it's a potential STQ site. So serine threonine kinases could be ATR, ATM kinases. Yep.
Channing?
So along those lines, Lou Cantley and colleagues recently redefined the sequence motifs for every serine threonine kinase, and it's now available as a database. Have you tried using that to see whether you can get a more accurate prediction of the kinase?
So the data suggests that U2F1 is phosphorylated is from a proteomics data. So we kind of has a idea which residue can be phosphorylated, but we have not applied a newer databases to see whether there's potential kinases that can phosphorylate that residue. But it's a good point that we should definitely look into that. Thank you.
That's a really cool paper, by the way.
So I'm looking at your model and I'm just wondering how you guys decided to pick the CRISPR-Cas9 as a screen, as opposed to just RNA-seq to figure out what changed, because it seems like depending where your guide RNAs are, you might miss splicing events that are critical.
Yeah, so this is just one part of the project. Of course, we are going to look into RNA-seq data of this engineered cell model, but CRISPR-Cas9 screen will allow us to identify the genetic vulnerabilities in these cells specifically. So I think these are two different approaches that we can take to focus on to study this project. Yeah.
Thank you. We should move on. Thank you. Very, very nice. We're right on time. 4 more to go. Our next is Jason Jones from Nick Levinson's lab. In search of the structural mechanism regulating CDK4 inhibitor interactions. Thanks, Jason.
All right, so I imagine a world where there's a precision medicine that can be used to treat any kind of cancer, period. The image behind me is an example of modern day precision medicine. So what you'll see is 2 proteins and a drug molecule. The magenta colored molecule that's kind of nestled inside of that cyan colored protein is called abemaciclib. Now, abemaciclib is one of 3 FDA approved drugs that are used to treat breast cancers, where an enzyme called cyclin dependent kinase 4, the cyan colored protein, is known to be involved in breast cancer progression. Now, when the drug interacts selectively with the active site of cyclin dependent kinase 4, enzyme activity is inhibited and it interrupts the growth of cancer cells. Now, unfortunately, patients treated with these drugs can acquire resistance, and even some patients are resistant right out of the gate. So it is important to continue to learn about the molecular details of oncogenes, such as cyclin-dependent kinase 4, so that more targeted therapeutics can possibly be developed. So in my so now going back to this image, just take a quick look at it and just appreciate that this is a static image of a very dynamic biological molecule, when in solution is very dynamic. So in my research, I aim to investigate the dynamics of cyclin-dependent kinase 4 to try to identify what I call structural microstates that can be targeted by therapeutics. So what is a structural microstate? So looking at this static image, just try to imagine in your head that brown helix that you see. Imagine that brown helix moving back and forth, twisting and turning and stretching. That red loop, imagine it like a noodle flopping around in biomolecular space, kind of doing its thing. And then finally, the lime green protein disconnecting from cyclin-dependent kinase 4 and diffusing off into the darkness. Now, all of these states that we can imagine, all these movement states, and then the steps in between these movement states is what I would consider a microstructural state. Now, I can actually measure and observe these microstructural states by putting on what I call molecular motion trackers, okay? But I can do that by chemically attaching these molecular motion trackers to the protein and then watching them in vitro using various biophysical techniques that we have available in lab. Now, these experimentally observed structural states can then be leveraged to develop high throughput assays where we can screen for molecules that specifically target these microstructural states and disrupt the kinase activity. So all in all, it's my hope that my research will lead to the development of new targeted therapeutics in the fight against breast cancer. And that's it.
Jason, that's excellent with one slide. All of the movements you got in there on one slide. Like, wow. Questions for Jason? Carrie?
So are you going to focus on mechanisms of resistance to these CDK4 inhibitors, and do they have distinct motions? Micro motions, is that the idea?
That's where the lack of knowledge is generally for CDK4 in particular.
So you're going to be examining mutants?
I'm really proposing, so it's not it's CDK4, it's not a mutant that's targeted. So my understanding in the breast cancer is that typically like the kinase can be overexpressed. And so it's more if we drug the cancer, because the cancer cell is running rampant.
Right.
But now my part is the drugs that are working right now only target that active site. But then there's there's so much more to this protein than just that active site. And so if a door is swinging open, I can stop it halfway because it has a certain structural state, and I can block it with a certain shape, if you will, consider like a molecule. I'm not answering your question, am I?
No, I'm just trying to understand the basis for resistance in your models and how your approach will combat that.
I think that might be the resistance in the cell, because after the drug is given, right? So I'm really drilling down to the biochemical aspect and looking at the protein just in a test tube right now, right? So that I don't know, the resistant mechanisms are a little bit above my field, like the cell biologist, I guess. But it would be cool to work with and develop these things and kind of maybe work with cell biologists to do some cool science too. Would be great.
Are there mutations in CDK4 that drive resistance? My impression is that you have other targets, other CDKs that seem to play a role, like CDK6, and also more recently, people have found that CDK2 is a compensatory activity, such that a CDK2, 4, and 6 inhibitory approach will overcome some mechanisms of resistance rather than mutations in CDK4. But I'm not aware of.
I'm not aware either myself. It's more like duplicated genes or overexpression of one of the components
Okay.
that's causing the unregulated cell cycle, because it's a cell cycle.
Yeah, I think you're right. So downstream events from CDK4/6 would be cyclin E goes up.
Yeah, it's very, very complicated, lot deeper. Thank you.
Thank you, Jason. Great. You get the prettiest slide, winner of the prettiest slide award. Our next speaker is Will Carnegie from the Tanya Friedman Lab, defining low Lin tunes signaling as a function of differential immune receptor clustering. Thank you, William.
Good morning, everybody. Thank you so much for coming out. Today, I'd like to tell you a brief overview of my project in Dr. Tanya Friedman's lab, looking at how the sarcomere kinase Lin potentially is tuned in its signaling through receptor clustering. And so the immune cell type that I focus my work on initially anyway is macrophages. And so as many of you likely know, macrophages are involved with clearing dead tissue, clearing infectious agents, but also involved in the wound healing process after an injury of sorts. And so in cancer, however, there's kind of this duality that's seen within macrophages. You can have a anti-tumor effect or a cell killing capability, but you can also have this immunosuppressive effect that has been characterized. And so understanding this duality is really the crux of my project. And so two receptors that are involved in this duality is CD16, which has been shown to be able to promote an anti-cancer or a cell killing capability. And the other receptor being CD32B, which has been characterized to promote a immunosuppressive or pro-tumor phenotype. And so what's really interesting where Lin comes into this is that both of these receptors are dependent on Lin signaling. And so how does this duality exist? One clue might be in the fact that you have Lin A and Lin B, which are two isoforms of Lin. And so maybe this is kind of our answer to this puzzle of how do we have these two different behaviors occurring through Lin, through these two different receptor types within macrophages. So that's one of the questions. The other question is how does the process of immune receptor clustering lead to differential signaling? And so this is a separate question that's been shown within the field, is that through cases of high or strong clustering of the immune receptors, you are able to induce an anti-cancer effect. In this example, I'm showing you CD16. Whereas in cases of low receptor clustering, you don't see this anti-cancer effect. Instead, you see a pro-tumor phenotype from the immune cells, from the macrophages. And so this is kind of the second question that I want to tease out within my work. And so how are we actually going to tackle this question is by refining this method that I'm working on within the lab, working in the lab right now. And so the first part that you can see is these beads that I'm going to use. And so these beads are how I'm going to be clustering the receptors. And so by using different bead sizes, you're able to modulate the receptor clustering that's occurring on the cell surface. And how are we actually targeting these receptors specifically is by using anti-CD16 specific or anti-CD32 specific antibodies separately to target each receptor pathway. And then on top of that, the lab has previously been able to develop Lin A knockout and Lin B knockout specific mice. And so I can then isolate from those bone marrow derived macrophages or BMDMs and then target each receptor pathway with only activity of Lin A or Lin B present. And so with that, I would like to define then the receptor dependent signaling profiles of Lin A and Lin B. I would also then ultimately want to define the relationship of signaling from these receptors with different clustering strengths and really using this to leverage Lin potentially as a tool to promote an anti-cancer phenotype. So with that, I take any questions and thank you so much.
Questions for William? Is there a particular cancer model that you guys are focusing on for which type of cancer?
Yeah, so once I define these kind of basic science mechanisms, I think breast cancer would be one that I'd like to go after. One, because the lab has those tools already at hand. But the other reason that I'd like to do that is because specifically these pathways of CD16, CD32 are ones that I think we could really go after in that way. But it's not just breast cancer necessarily. It could be expanded to others, including like colorectal cancer as well.
Are LINs modified, overexpressed, or mutated? Did you say that in cancer?
So yeah, so that's a great question. I should have highlighted that more. Is so LIN overexpression is associated with worse prognosis, but what we don't know is the LIN A versus LIN B role that's being played there, and that's really something that's very understudied within the field overall.
Excellent. Questions for William? Scott?
Are LIN A and B equivalently affected by pharmacological inhibitors of LIN kinase?
I don't know that. I would guess that the mechanisms are likely the same. However, again, which one you kind of would want to promote signaling through is kind of a conundrum. So yeah, I see what maybe your question is when we target one, are we targeting both? In which case, are we losing potentially positive associated factors? So I would say yes, they likely are targeted the same way. The only difference is this 21 amino acid insert, and that's not where you would expect any of the inhibitors to be targeting.
Excellent. Jaime?
So you're doing this in mouse models, right? So I'm pretty sure the distribution of a lot of these myeloid receptors, including 16 and 32B, is different across species. But do you know if the signaling remains? In other words, even though different myeloid subsets express the proteins at different levels, is the signaling pathway for each one of those conserved across species? Or is that going to be a very mouse-specific finding?
So in the context of these two receptors, they should be conserved across the different species. One that is kind of a tricky thing is so there is a CD32A, and mice lack that receptor, whereas humans have that. And so that would be something that expanding this work into other immune receptors, we could get into some kind of tricky business there. But in the context of these receptors, is why I chose these ones to look at their contribution specifically. Yes.
Excellent. We should move on. Thank you, William. It's beautiful. Next, we have Megan Ludwig from Justin Drake's lab using the extracellular vesicle proteome to understand therapeutic resistance in advanced prostate cancer. Thank you, Megan. Thanks. So my project focuses on extracellular vesicles, or EVs for short. These are nano-sized particles that cells export for communication purposes. They can be found in every biofluid, such as blood and urine. Given their high numbers as well as stability, they have great potential for being developed into liquid biopsy tools. And so we're hoping to use that in advanced prostate cancer cases. So here, what I'm showing you is the line of a tumor growth, where a patient can respond to local therapy as well as androgen deprivation therapy. However, resistance can occur. And so there are multiple mechanisms. One being the cells can still rely on androgen receptor signaling, such as the expression of AR variants. We also have those that express neuroendocrine features or double negatives. So they're no longer reliant on either AR or the neuroendocrine pathways for survival. We hypothesized that EVs could distinguish between these different subtypes. We focused on the proteins that these EVs carry as cargo, because proteins are a more direct measurement of cellular activity. So we took the proteins from EVs that I isolated from multiple prostate cancer cell lines and then characterized the proteome by mass spec. And as you can see from this heat map, these EVs did actually cluster by subtype. So the blue here, these are the EVs isolated from AR positive models, green is from neuroendocrine, red double negative. And then we also have the interesting case of LNCAP95s, which are cells that express AR, but the EVs are more related to the double negatives, which is still an interesting special case that we're trying to figure out. But given the thousands of proteins that we identified in these EVs, we've been able to use traditional gene signatures, such as looking at measures of AR activity. We've been able to look at cell type markers, such as those expressed in neuroendocrine cells, as well as do gene set enrichment analysis, and found that EMT proteins are enriched in the double negative subtype specifically. And so we're hoping to use all of this information that we are able to find in the proteins of these EVs and match that with functional role that EVs have in promoting tumor progression. So EVs have been observed to have a role in treatment resistance, proliferation, as well as promoting metastasis. And so we hope to identify specific EV proteins that are related to these functional phenotypes to better advance liquid biopsy tools. So overall, our results support the fact that EVs have a lot of potential as developing liquid biopsy tools, and especially in increasing the sensitivity of EVs and looking at EVs specifically derived from tumor cells in order to successfully advance liquid biopsy tools, make, for example, a blood or urine test for patients to make it easier and quicker to check on tumor status than, say, a tissue biopsy. So with that, I'm open for any questions. Thank you. Great. Questions for Megan, Carrie?
So EVs are comprised of a bunch of different size vesicles, right? Exosomes and
Yes.
like that. So
Yes, you're right.
are there any differences in these different prostate cancer cell types in terms of the proportion of exosomes to larger vesicles and things like that?
That's a very interesting question. Since we can't necessarily look between, say, exosomes versus microvesicles and that overlap, it's difficult to say since we can't isolate those populations. From what I've seen from looking at the sizes, they are overall fairly similar from the population that I'm
So the
isolating.
distribution of sizes is similar.
Yeah, yes. Question for Megan in the back.
Do you think you can gain insight into a man's like tumor heterogeneity from these EVs, where some might be more ARPC and you might see some increases in like NE markers?
Yes, I think so. I think it would require, like we would see, for example, like both markers from the two different clones, and they would probably be together. Analyzing EVs on a singular scale, kind of like a single cell analysis, would be very difficult, I think, at this point. So it would be limiting to the exact, like how many clones and that kind of a thing, I think, yes, but to some degree. Are you thinking we're moving towards a world in which they can be a paired diagnostic, maybe something like that? Yes, Scott.
So the proteins that you're discovering in your EVs, are you finding them to be enriched for proteins that have certain subcellular localization? I would guess nuclear proteins probably aren't really present in the EV proteome, but I may be wrong.
No, you are correct. Yes, nuclear proteins are not very enriched. It's mostly membrane and cytoplasmic proteins. Yep. Cool. That was great. Beautiful. Thank you, Megan. Our last speaker is Alexei from Ingenström's lab. Pathways to utilize a novel transgenic TCR murine model for pancreatic cancer immunotherapy. Thanks, Alex.
Thank you. All right, in Ingen's lab, we look at a lot of T-cell based therapies, and these cellular therapies have become prevalent in the treatment of various hematologic malignancies in humans. And studying them and using models in mice is challenging because any of you who have worked with primary mouse T cells know that they're fickle and they don't like to be manipulated and they don't like to live for a long time outside of the mouse. And altering them in any way is very challenging. So to try to produce a model that has more reproducibility, that has a little bit more consistency, that we can manipulate easily, a while ago, one of the graduate students in our lab produced a really unique mouse model in which we took a couple of TCRs that we had previously identified, so T-cell receptors that are specific to a tumor-associated protein called mesothelin. And we use CRISPR-Cas9 to insert that into a very specific location in the mouse genome called the TRAC locus, or the TCR alpha constant locus. And what that ultimately results in is a mouse that produces almost a pure population of our sort of specific TCR expressed on CD8 T cells. And not only that, but this TCR is regulated in a sort of normal manner, which you don't get when you use sort of a traditional retrovirus transduction, which is what we would typically use in the past. And so you can imagine that we can use this mouse model to do a variety of things. And so I've just listed, I don't have any data today that I want to present, but I just listed some questions that we have or are considering pursuing. And the first is easy. So we have two different TCRs that we identified and sort of put into these mice. One has a high affinity for this mesothelin molecule. The other one has a low affinity. And we can assess and see whether or not, a stronger TCR affinity actually is better or might have some negative consequences. We can look using these T cells at how sort of adoptive transfer and therapeutic models can work in different models of cancer. So not only pancreatic, which we focus on, but other models like ovarian and lung, where we see mesothelin expression. And then we can do further things to these cells now. We can alter other genes. So just as an example, you could knock out other proteins like PD-1, for example. But it's not just the therapies that we can study. We can actually look at very basic questions. And so on the bottom here, we could look at tolerance and how these cells are not killing the mice, because in fact, these mice have mesothelin expressed on some of their normal tissues, and these mice don't die, they do fine. And so why is that? That's probably due to a concept that we call peripheral tolerance, which is very understudied still, okay? And we can also just look at the development of these T cells. This system has sort of been hijacked, and we can just kind of follow along and see how these T cells are developing, how they go through another process called central selection, and how, you know, if there are any sort of changes that are happening in our model. So this is a powerful tool that we're now using a lot in the lab to answer these and more questions, and also, you know, to sort of help to develop further advancements in the cellular therapy field. So with that, happy to take any questions.
Great, thanks, Alex. Questions? Jaime has a question.
So this is a really cool model. You mentioned peripheral tolerance, but in the mice where you're actually putting in the transgene, you know, I'm assuming this is now carried on the line, right?
Yep.
So does central tolerance play a role? Because I think being able to do adaptive transfers would give you a really good idea of the toxicity profile of the cells.
Yeah.
In modified after birth, right? But there must be some central tolerance.
There is. So if you examine these mice and you look at mice that have or do not have mesothelin, for example, you will see that the mice that are, you know, the T cells that are raised in mice that have wild type mesothelin have fewer CD8 T cells. Some of those T cells are dying through central selection for sure, but there are many that escape that process. In fact, there are plenty of CD8 T cells still in those mice, and they don't cause havoc, probably because of peripheral tolerance then. So yes, that process does happen. The affinity between
the cells that escape central tolerance.
this is, yeah, so this is generally a pure population. They only express one TCR, which has a set affinity for mesothelin. So, yeah. We haven't studied that exactly. There may be some differences in, for example, TCR expression too, the amount of TCR that makes it out onto the T cell surface, but we haven't seen that. There's still very high expression. We haven't tried to answer that question very specifically though.
So you mentioned that one aspect of your studies is look at pancreatic cancer, but what you described here, it's hard for me to envision how you're going to combine those aspects. Are you going to use pancreatic cancer mouse models and then manipulate the T cell receptors for high and low to see how pancreatic cancer responds in that setting? Or in other words, how are you studying pancreatic cancer?
Yeah, so we already have answered or tried to start answering that exact question. We have a very established model of orthotopic implantation of what we call KPC cancer cells that are placed into the pancreas. And then we perform adoptive transfer. And we've compared the sort of 1045, the high affinity and the low affinity TCRs in preliminary analysis. And there are some differences. And so that's how we study this, is sort of through a model of adoptive transfer. Yep.
That's a great point. I was going to ask the same thing. Where's the cancer? Where's the cancer? Anyone else? We are right on time, miraculously. Can everyone please give a round of applause to Alex and all of our trainees? Thank you so much.